Hello, my name is Juliana Giles, and this is my presentation for this semester's Experimental Design and Statistical Analysis class here at Columbus State University. The title of my presentation is Understanding the Effects of Varying Salinity Treatments on Euca Species Within a Freshwater Tidal Marsh. So I want to give a little bit of background information about freshwater tidal marshes before I dive into the analysis that I chose to do. These systems are already naturally resistant to fluxes in environmental processes, and that makes sense when you consider where they're located at that threshold between ocean and land. But the biggest threat that they face today, especially, is the loss of the vertical buildup of sediments that gives rise to the literal surface area of these habitats. And the reason why the vertical accumulation is at threat is because of continuously rising sea levels. So not just a flux in sea levels or a flux in freshwater discharge that would produce a small response, but continuously rising sea levels that's really going to impact the ability of that system to accrue sediment over time. So this would lead to a change in habitat area, a change in the salinity and the freshwater influx, so a change in the literal chemistry of these systems, and that's going to lead to a pretty major biological shift. So researchers are currently focusing on monitoring the long-term effects of increasing salinity at these long-term research sites in order to get a better understanding of what kind of community shifts we're going to be seeing if sea levels continue to rise on their trajected paths. So one of those long-term research sites is actually the focal point of the study. It's named SALTEX, which stands for Salinity Addition Long-Term Experiment, and it's located in Champney Island, Georgia, along the Altamaha River. And the overall goal of this long-term experimental site is to observe and better understand the effects of saltwater intrusion on a low salinity freshwater tidal marsh. And the methods that are deployed at this research site are cited in a ton of publications, but I included the citations from two major publications that talk about the methods. Essentially, this research site is subdivided into experimental plots. These plots are treated with different types of water treatments, and they're really interested in understanding the press and pulse salinity treatment plots. So in the press salinity treatment plot, that area receives a constant increase in salinity, and that is so that the researchers hopefully can gain a better understanding of how these types of systems are going to respond to in continuous increase in sea level rise. The pulse experimental plots are going to simulate a response to a periodic or episodic influx of salinity, which could be caused by drought upstream of the freshwater source. So in this case, the Altamaha River, and that can be caused by changing precipitation rates as a response of global climate change. So just for a little bit of context, I included a map from Google Earth that shows you the SALTEX plot in conjunction to the Altamaha River. And I've also listed the 5 experimental treatment plots that the SALTEX research site area is subdivided into. So if you look in the top left-hand corner of the screen, you'll see those 5 sites are listed. So the first 2, the pulse and press salinity treatment plots, I just talked about those briefly in a previous slide. There's also a freshwater addition plot, as well as 2 control plots, one with sides and one without sides. And this is just another map from Google Earth that shows you how close the SALTEX plot is to the Atlantic Ocean. So now it's a little hard to see here, it's pretty zoomed out, but if you look right here where I'm circling in red, that's where the SALTEX research site is, and you can follow the Altamaha River all the way down to the Atlantic Ocean. So the data that I analyzed in this study came from the Georgia Coastal Ecosystems LTER online data hub. And I used a data set that that was published by a team of researchers led by Christine Angelini, who is with the Department of Environmental Engineering at the University of Florida. This data set was made publicly available in 2018, and the title of the study was Effect of Saltwater Intrusion on the Distribution of Invertebrates in a Georgia Tidal Freshwater Marsh from the GCE Saltex project. The invertebrates that Angelini and the rest of the researchers that conducted this experiment chose were Uka species or fiddler crabs, and they were not specific in the species of fiddler crabs that they were observing. I'll talk about that in just a moment, but I did include some pictures of the 3 most abundant species of fiddler crabs in Georgia. So on the left you have the sand fiddler, in the middle you have the marsh fiddler, and on the right you have the red-jointed fiddler. So these are very handsome creatures. They have that characteristic big claw. These are males, and that big claw is how you know it. So Angelini and the other researchers that worked on this data set determined fiddler crab densities based off of the number of burrow hole counts within those experimental plots. They accounted for adult burrows and juvenile burrows as well. So the data set was organized into adult counts and juvenile counts. Adult counts were considered to be anything that was greater than 0.5 centimeters, and juvenile burrow holes were considered to be anything less than 0.5 centimeters. In order to be counted, these burrow holes had to be circular in shape, and they also had to have evidence that they were being actively maintained. And the reason why they chose to count burrows instead of individuals is that these guys are typically in their burrows during times of high tide. They only really come out at low tide, and they're crabs, so they're pretty elusive. It's just easier to count by burrows. So that's how the data was collected within this data set. So I've included several pictures here that are just fiddler crab burrow holes. This is what they were looking for in that Saltex freshwater tidal marsh. And you can see some more evidence of that big male claw in that bottom right GIF that I included. So my goal for this study was to organize and or aggregate data if necessary for analyzation. The question that I set out to answer was, do the experimental treatment plots at the Saltex research site influence the observed number of fiddler crabs within the plots? And the hypothesis that I proposed was that there would be a significant relationship between the experimental plots and the number of fiddler crabs observed. Right, moving on into my methods. The first thing that I did was acquired the data set from the Georgia Coastal Ecosystems LTER data hub. So I have included a screenshot of what that data looked like when I got it from the website. So you can see here the data organized by the sampling day. They have the experimental treatment plots listed. They have the adult fiddler crab burrow counts as well as the juvenile fiddler burrow counts. So this is the data that I was really interested in, and I had to aggregate that data to make it more meaningful for me to analyze it. I began organizing the data from the original data set based off of sampling date and the experimental treatment plot. So if you look right here in this yellow column, these are all of the treatments. There were 8 individual sampling dates for each of the experimental treatment plots, and for each of those days I took an average of the adult as well as the juvenile fiddler crab burrow counts. That gave me 40 individual replicates to analyze. So before I began the analysis in JASP, I did a little bit more organizing. I assigned numeric values to the 5 different treatments, as well as the adults and juveniles to distinguish whether the counts belong to either or. And I did that just so that the analyzation would go a little bit smoother in JASP. I decided to run a 2-way ANOVA for this data. And that was just because I have 2 factors, which are the different treatments and the life cycle status, so whether they were adults or juveniles. And these are the results from the 2A ANOVA that I ran. So this is the ANOVA table. you can see that there was a significant effect of life cycle status on the mean number of fiddler crab burrows that were observed. But there was no significance among the experimental treatment plots, and there was also no significance within the interaction term, so the adult or juvenile status and the experimental treatment plots. I did run an assumptions test. I used the Levine's test for equality of variances and found that the data were actually in violation of that assumption. So the variances were actually heteroscedastic. But if you look at this descriptives table right here, you can see just the amount of variation between the adult counts and the juvenile counts themselves, which is likely what caused the data to be in violation of that assumption. So I ran a Kruskal-Wallis test, which is a non-parametric test similar to a one-way ANOVA, and it produced similar p-values. So I continued on with my analysis, even though the data were in violation of that assumption. This is a graphical representation of the results. So even in this graph, it's pretty obvious that there is a significant effect on the mean number of burrows observed between the adult and juvenile fiddler crabs, but there is no significant effect between treatments, and there is no significant interaction effect between the treatment plots and the number of observed fiddler crab burrows between adults and juveniles. So wrapping it up, just to remind you guys, the purpose of my project was to determine whether there was a relationship between the Saltex research site's experimental treatment plots and the densities of adult and juvenile fiddler crabs. However, my hypothesis that these treatment plots would have a significant effect on the mean number of burrows observed between adults and juveniles was not supported by this analysis. However, I do think that it's important to note that that was not the point of this data set. That was just the question that I wanted to ask about this data set. In the future, if there are further invertebrate studies conducted, it probably would be more helpful to answer a question like this if there was a more robust sampling techniques. Maybe if they sampled on more than 8 days per plot. However, I do think it's important to just make one final note that there's lots of really cool research being done at this site. There are lots of insights from other projects that can give us a good glance into the future as to what we can expect to see in terms of biological community shifts in response to saltwater intrusion. These are the references that helped me formulate my project proposal as well as assemble this presentation. So before I wrap this up, I do have a few acknowledgments that I'd like to make. First and foremost, Dr. Troy Keller, thank you so much. Your advice and expertise is invaluable, as always. I also want to thank the Georgia Coastal Ecosystems LTER Data Hub for making these wonderful data sets available. Christine Angelini and the other researchers who contributed to this data set. I'm very thankful that this data was even collected. And I'd also like to thank you guys for making this such a fun and enjoyable semester despite the strange circumstances. I look forward to watching your presentations, and I hope that you all have had a wonderful semester and that you have a wonderful winter break.